Hi, this is Dr. Lauren Launen, and this is part 2 of an online lecture aimed at introducing you to the genomics approach utilized in the Vibrio vulnificus project happening in my research lab at Keene State College. So I left off talking about sequencing, and we use Illumina high-throughput next-generation sequencing, and we get 250 base pair paired-end reads out of that sequencing. And I've talked about this before in class, but what that means is you take genomic DNA from a pure isolate and you fragment that DNA into smaller pieces, and then you add adapters and other important sequences to the ends. You apply that, what's called a library at that stage, to the flow cell, and that material is going to bind to the flow cell. You use PCR to do a technique called bridge amplification, which leaves you with little clusters of identical sequence. And then you actually do the sequencing, and that's done with a reversible dye-terminator reaction that I discussed previously. And so as that happens, we'll see flashes of color where the correct nucleotide, according to the template sequence, is incorporated into the growing strand. And so what this results is the creation of a very large data set where many areas of the genome might have been sequenced multiple times, hopefully 50 to 100 times in fact. That's really good coverage. We take that data and we process it by removing the ends, and we use a specific program, computer program in order to do that called Trim-O-Matic, and then we take that data and we run it through an assembly using spades, and we run a quast report on it to see what the quality of the assembly was like, and we run a program called Proca on it to annotate the genome. Another thing we do with that data is we take that raw Illumina data and we do a technique called MLST, and I'm going to talk about that next. So MLST is a classic methodology used in microbiology, and it's used a lot in medical microbiology to type different members of or different strains from the same species. So multi-locus sequence typing, or MLST, involves analyzing sequences of selected genes as a group. And the particular scheme showed here was developed by Dr. Niall Bisharat, and the information for it is maintained at this public database or resource. But Niall Bisharat determined that you could use these genes, GLIP, GIRE, MDH, METG, and PER-M, all from chromosome 1, and these genes, DTDS, Lyce-A, Pint-A, Pyr-C, and Tra-A, from chromosome 2. And if you could get the sequences from these 10 genes, and then study them by concatenating or grouping them together, you could do phylogenetic analysis, and you could also assign your isolate to see if it matched anything previously published or documented. So you could assign it what's called a sequence type. So sequence type 1 in this example has allele 1 of the GLIP gene, 5 of the GIRE gene, 54 of the MDH gene, 23 of the METG gene, and 8 of the PER-M. These numbers are a catalog of alleles for those particular genes, or from a catalog of alleles for those particular genes that's maintained at this site, and that was developed by Dr. Niall Bisharat. Similarly, if you're sequence type 1, you've got allele 4 of this gene, 13 of this gene, 26 of this gene, 71 of this gene, and 32 of this gene. If you have different combinations of allele, you're a different sequence type. And so doing this kind of analysis, we can see if our isolates from the Great Bay Estuary are the same or different from anything previously documented. And we can also contribute novel sequence types as we discover them. What those particular genes encodes is given down here. So we've used Niall Bisharat's scheme, but we've also created our own scheme by looking at these genes, the 16S rRNA, which is a phylogenetic marker gene, and there are 2 different types commonly found in Vibrio, and then these genes here, all of which encode pathogenicity factors. So the V is for virulence in this case. So I'm going to draw your attention to WZA, B, and C. These are genes that must be expressed to make proteins that will enable Vibrio vulnificus to make a capsule. And capsules are definitely virulence factors, as you should remember from learning about Fred Griffith's work in Streptococcus pneumoniae. So the basics of MLST is that you've got known alleles of a particular gene in a reference database. We're using either the common 10 housekeeping gene one developed by Niall Bisharat, and we're also using a reference database that we've developed ourselves for Vibrio vulnificus. You take the reads from Illumina sequencing and you map them against those alleles, and hopefully you get high coverage because low coverage doesn't give you good data. And from that you get a consensus sequence, which is the sequence for that, for that particular gene that you're studying. We use a computer program called SRST2 in order to do multi-locus sequence typing. And we've been doing this for a while, and we've got some pretty interesting information. The consensus sequences for 16S rRNA have been used by Sarah Sanders to generate phylogenetic trees. And if you'll remember, one of our hypotheses was that the isolates from the Great Bay Estuary are not pathogenic, that they're going to be more like environmental strains. And this phylogenetic tree actually backs up that hypothesis. It lends support to it. All of these are Vibrio vulnificus from our sequences of 16S from Vibrio vulnificus isolated from the Great Bay Estuary, and they're grouping phylogenetically in a different clade. So this is a clade, and this is a clade. And this clade down here has 2 known pathogens that we work with as reference strains. So we got that information as an offshoot of doing MLST work. Sarah also used our custom database, and she obtained sequences for the different virulence genes and also the 16S gene, and she concatenated or grouped them together into one long sequence and did phylogenetic analysis. And what came out of that, and there are no reference strains in this, but what came out of that is that our isolates group into 2 major clades, this one and this one. And it's not random who goes where. It looks like the 2015 isolates are grouping phylogenetically quite differently from the previous isolates. And if you'll recall, earlier I mentioned that there was some indication from the numbers of isolates recovered per sampling event that there might be an expansion of some sort of Vibrio vulnificus that is easier to culture and therefore provides more isolates per sampling event. This data, this phylogenetic tree, really does suggest that there is something different about the 2015 isolates, that they're changing, they're responding differently, and they're genetically different than the pre-2015 isolates. So we're interested in following up on that work. That's the MLST work and where we're at with it so far. The project that, if you're in Bio 311, you're actually working on is contributing to this workflow, and that's whole genome analysis and comparisons. So in that work, you take all of the Illumina data, and after you've cleaned it up, and you assemble it, and from that you get what are called contigs. And only one contig is shown, but an assembly should actually provide maybe a few hundred contigs. You then take those contigs, which are long DNA sequences that are hopefully similar to what was actually originally found in the organism, and you annotate it. And annotation is done using PROCA in our pipeline. Assembly was done using SPADES. And annotating means you use a computer program to go through and look for genes. So this little blue line here is a gene, and this little red line is a gene, and this is a gene. This is a gene, this is a gene, and so on. And then that particular sequence is submitted to the NCBI and other resources, and they're named so that we know what this gene is, and 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 so on. The pointiness of the arrows indicates the direction in which transcription occurs, by the way. Now, if you do this using PROCA on all of the isolates, and so far we've done about 60, or we're working on about 60, we should have 60 more from the 2016 data set that we work on over the next year, then you can compare what you got between isolates, and you can build figures like this. So each line, blue, green, and red here, represents chromosome 1 or chromosome 2 from one particular isolate of Vibrio vulnificus. And this is from a publication. This is from previous work, not from our Great Bay Estuary work. We're not quite at that point yet. But you can see if they have common genes or different. So where colors line up, they have common genes. Where there are gaps, one organism might have that and another one might not. So we're able to, if you were to zoom in on this, you would be able to see similarities and differences, what a shared core genome was and what some isolates had that was unique from other isolates. This is where we're at so far, and this is after working on this project for about a year and a half. We've got methodology for the MLST analysis, very well established. We're pretty good at doing that now, and we're actually using that methodology to generate results. For example, we're seeing that there are a lot of novel sequence types, different Vibrio vulnificus than have been found elsewhere, and we've also seen the emergence of a new sequence type in 2015 that wasn't previously in any of the samples. And we've been able to refine our MLST methodology, and we're still working to do that. So we're trying to add new pathogenicity associated genes, refining our custom database. We're going to contribute alleles for the 10 housekeeping gene scheme to pubmlst.org so that we're sharing our work with others, and that there should be a lot happening in that area over the next year. We're still really aggressively working on getting the methodology for whole genome analysis operating. So we're pretty good now at assembling and annotating and evaluating single genomes. Next, and that's what you're doing in the class project. Next, we want to be able to take that and start using a program called rRNA that allows us to do the comparative genomics that I mentioned on the previous slide. And we're also implementing this work as part of class projects. So we're excited about having students in class actually contributing to the methodology, practicing with it, and ultimately using it to generate data that's actual research that you can do in a class setting. We're also working, outside of this genomic stuff to analyze the environmental data that has been collected through the Vibrio monitoring program, and that is what has shown us that there's the expansion of a strain that is more culturable in isolates coming from the Great Bay Estuary in recent years. And so we're going to be using that data to link it back to the genetic data that we're getting in the lab. Going forward, we want to be able to design appropriate experiments where we can look at a phenotype of interest like temperature tolerance, growth rate, and we can link it back to the presence or absence of virulence factors, and see if we can also maybe develop some rapid screening methods so that you could quickly look at a sample from the environment and see what Vibrio vulnificus is present, and if it's similar or different from what was previously present in the Great Bay Estuary. So again, I'll acknowledge that this work is not done in a vacuum, that there's actually a large team of people doing this work, and funding sources for those people. And I'll just draw like a special shout out to the student researchers. Sarah Sanders, Katie Kiley, Rick Pereira, and Caitlin Smith have worked so far on the project, and we've got a new gang coming on board soon. If you're interested in this kind of work in future, let me know. From time to time I have a spot in my lab.